let's start.
Okay.
Talk a little bit about the whole field of liquid biopsies, if you want to begin with, and then how is Epic approaching this?
Okay.
The whole concept of being able to go in and just from a patient's blood, detect cancer and other diseases.
Yeah, and the field of liquid biopsy represents meeting a way to meet a lot of unmet medical needs for patients. So it's very hard in some of these late stage patients where they have multiple metastatic sites, multiple metastases. They've all gone. They have tumors, multifocal polyclonal disease. That can be very hard to take a biopsy and discern anything from. So liquid biopsy represents a chance to rather than go in and drill out a bone metastasis, that I can take a simple peripheral blood draw and get the same information or get unique information that I miss by biopsy.
Now, there are very few circulating tumor cells, right? So you have to get those, maybe a dozen or so out of millions.
Yeah.
And so how do you do that?
And so really the interesting part about Epic is we've taken a very simple approach. Traditionally, people have taken the approach of, well, let's enrich, let's separate the tumor cells from the white blood cells. Whereas Epic, actually, we're looking at all nucleated cells. So we take a simple blood draw, we get rid of the red blood cells by lysis. Every cell, every nucleated cell gets put onto glass slides and we image it. And so we let the imaging figure out which tumor cell it is.
So how many cells are scanned in a typical assay?
So typically we look at about the equivalent of about 1 ml of blood, but that can vary based on the clinical question we're trying to answer.
Okay. And so how many cells are typically in those?
So typically a slide holds about 3 million cells,
Okay.
and we make about 10 slides from a tube of blood.
Obviously you're not looking at these manually.
Right.
You have to have some kind of software and interpretive system to characterize them.
Yeah, really the driver of this is we've developed a very nice imaging system that can rapidly look at cells not only for fluorescence intensity but also for morphology, size and shape, how they're different. So basically the algorithms behind this are similar to what are used in facial recognition and other things, where we're looking for rare events. So you have 3 million what are very normal white blood cells, and surrounded by a very small number of tumor cells in some patients. And so the algorithms find the rare events with great statistical precision because we have 3 million normal controls on the slide.
Okay. All right. So once you've got those cells, what do you do further to see what they represent? Do you look at their genome at all?
So we start by looking at protein expression and morphology, but then we can, for each cell that's on the surface of the slide, after we assay it for its morphology, its physical characteristics, its phenotype, we can extract that single cell from the surface of the slide and we can look at its genotype. And so that's just single cell sequencing. So we've developed a lot of techniques here that are specific to focus on just sequencing DNA from single cells.
Okay. And what the whole idea of liquid biopsies has had a lot of bad press because of Theranos. How do you and other companies address that? Because that stuff has been very much in the news. And how do you say our technology actually does what it's supposed to do?
Yeah, and that's a great question. And at Epic, one of our key focuses is not trying to we build tools, which then we build assays to answer specific clinical questions. And so clinical validation is incredibly important to every one of our tests. So actually demonstrating in a patient cohort that our test does what it's supposed to do.
Okay. What are some of the most common tests that you're asked to run?
So we do our main test that is focused in prostate cancer. It's ARV7. Okay. It tells a patient with metastatic castrate-resistant prostate cancer whether they should take an AR targeted therapy or they should be switched to a chemotherapy, which is a much cheaper and they'll live much longer on that.
Okay, and so what are the things you're looking for that help make that decision?
So the way we've always focused on clinical outcome. So when we develop tests, we look for ARV7 as an example. We look for the signal of the truncated ARV7 isoform within the nucleus of a single tumor cell, and that's a positive. And so the difference being is we're focused on the clinical outcome, so we know that that signal has a specific clinical utility.